And this document is available to you. I already posted it on the Google Classroom. So it's on my Google Drive in the folder, and it basically is something I did a couple of days ago where I just typed up everything I could think of that we went through over the course of the year. For us, it was really only 5 units. For the College Board, they consider the 6 units because they consider signal transduction like a separate unit, which is fine because it is really kind of, a lot of stuff and it's hard, and I threw it into cells, whatever, it doesn't really matter. So I split it up here as a separate unit so it matches the College Board 6 units. All right. So having said that, I'm going to race through this and I'm going to talk 100 miles an hour. I'm probably going to need to take a break and get another water bottle because my throat gets really dry when I talk a lot. I only have this much left, so I may be getting up and leaving in the middle of this. But okay, if you have questions as I go along, like if you come up with something that you want to ask about, just shout it, just turn on your mic and shout and stop me and whatever. Please do, like, interrupt me and stuff, that's fine. Please don't be scared and don't be shy and stuff. Like this is crunch time now. We have less than a week left before the test, and so if you have issues that you want me to go over, please say them. Otherwise, I'm going to start now. All right, our very first unit was biochemistry. As a matter of fact, we started with like just like regular like inorganic chemistry, where we talked about water in chapter 3. So the important stuff to remember about water is that water is obviously H2O, and it is polar because if we look at a water molecule, it looks like Mickey Mouse. It's got the 2 hydrogens and it's got the oxygen, and the oxygen is highly electronegative, which makes this part slightly, the chin is slightly negative, and this is slightly positive over here. So we say that's polar. And the fact that water is polar, it has the properties that it has, like affects just about everything we do for the rest of the year. I mean, we talk about that and talk about how membranes are nonpolar and hydrophobic, again and again and again. So that's kind of an important thing. So you should know about water, and obviously water forms hydrogen bonds with other water molecules and with other partially charged molecules. So hydrogen bonds are just simply the bonds between 2 partially charged regions of a molecule, and it doesn't have to be water, it could be other molecules, but generally a hydrogen atom is involved. So so that stuff is important. We talked a bit about carbon compounds, and I made you like learn the 3-methyl pentanes and all that crap. Don't worry about studying those. I just did that because I'm evil. But you should know that carbon has a valence of 4. That means it likes to have 4 bonds associated with it. So carbon is going to have, always 4 things like this because of its valence of 4. And then when we do look at carbon compounds, there needs to be 4 bonds. So a lot of times that means that there's 4 other atoms stuck to it, but sometimes there could be a double bond like this, where like C double bond O, and then there'd only be 2 other things stuck to it. Like in the case of an amino acid, for example. The carboxyl end would look like, well, okay, I wrote this backwards, it'd be HO. And in case you don't remember, the whole idea about the HO and OH deal is that you have to keep the valence of each of these things in mind. And you have to remember that oxygen can only bind 2 covalent bonds and hydrogen can only bind 1. So if you're writing a compound like this, it really should be HO instead of OH, because the carbon is connected to the oxygen and then the oxygen is connected to the hydrogen. The oxygen is the connector, the hydrogen cannot be a connector. So that's important to remember. And obviously then the rest of the amino acid would look like this. Okay, and we'll talk more about amino acids later, and that'll come up again. Obviously, you know this already. This is the part where you nod and make me feel good. All right, so then we jump into biological molecules, and there's 4. There's proteins, there's carbohydrates, lipids, and nucleic acids. Can you just quickly turn off your mics and everybody together, like a choir of angels, shout out which of the 4 is the most important?
Proteins.
Yay, that was a choir of 1 angel. I love it. Okay, proteins, yes. Some people argue DNA is the most important, but my argument is that the pie is more important than the recipe. Yes, you need the recipe to make the pie, but the pie is what you can eat and use for food and all. So anyway, let's start with proteins. Super important. We'll talk about them first. They're made out of amino acids. So here's the structure of an amino acid right here. It's got a carboxyl end, it's got an amine end, it's got a central carbon, it's got an R group, and it's got this one extra little hydrogen. Are you able to see my cursor? Are you able to see this? Okay, so the R group is the most important. Everything else is like the same on every amino acid, those parts are. And then the R group is what makes them different. And there's 3 different kinds of R groups. There's polar, there's non-polar, and there's charged. And that was actually on one of the practice questions. I kind of was surprised they would ask that, because that's kind of like a memorization thing. And usually they don't ask too much memorization stuff. Anyway, okay. So polar just means they have slight charges on them. So in other words, they have like oxygen or nitrogen in the R group. Non-polar means it's just carbon and hydrogen mostly, meaning there's no electronegative parts at all, which means there's no partial charges. And then charged would be like the acid and the base ones. All right, and the acid ones would be then negative and the base ones would be positive. All right, so amino acids or proteins have different levels of structure. Primary structure is just simply the polypeptide chain. It's just a chain of amino acids linked together with peptide bonds through dehydration synthesis. Just a bunch of them. You should be able to, draw 2 amino acids next to each other, and you should be able to see how H2O pops out and then how they link together. And obviously this hydrogen right here comes out. Oh, I'm sorry, this whole OH comes out and this hydrogen, and then this would link up to the next carbon up there. But that's, that's like freshman stuff. You guys know that. So that's primary structure. Secondary structure is then when those chains, those polypeptide amino acid chains fold, and they can fold either into alpha helices like this, or they can fold into beta pleated sheets like this. And I don't draw the pleats, and we don't draw the pleats when we draw out these sheets. You do not need to memorize the structure of a beta sheet. I made you learn that for my test, but you don't need to for the AP test. And what you should know about these is that they're held together by connections between so hydrogen bonds between slight negative on this oxygen and slight positive on this hydrogen. Now, obviously, this one would match up with the double bond O up here from a different molecule, and this would match up with a hydrogen from a nitrogen up here. That's slightly positive. So these are the hydrogen bonds that form, and it's just connections of hydrogen bonds between atoms of the backbone, not R groups. No R groups involved in secondary structure. So alpha helices, beta sheets, these are not to be confused with the helices that form with starch, with carbohydrates. These are completely different. These sheets should not be confused with sheets that form with cellulose. Totally different. Just happens to be called sheet, happens to be called helix. Don't confuse them. Tertiary structure is then when these guys fold some more. And now we have stuff like this. We have a beta sheet here. We have an alpha helix. We have some more folding. We have another beta sheet here. And this folds into a complex 3-dimensional globular structure. And tertiary structure is held together by a bunch of different connections. So, okay, maybe this is a bad drawing. Let me do a better one here. So let's say we have, a beta sheet here. We got some alpha helices here that twist around. We got some just normal kind of stuff here, maybe another beta sheet. We might have a connection between our group here and here, connecting these. And that could be covalent in case of a disulfide bridge between 2 cysteines. That could be hydrogen bonds. Maybe over here we have a hydrogen bond between charged regions. It could be an ionic bond where, if this comes around here, maybe we have an ionic bond between this area and this area where there's a plus here and a minus here. All of these different things are possibilities. So disulfide bridges always happen with cysteines, 2 cysteines. They have those sulfurs in them, and they make a sulfur connection, which is super strong. Oh, I don't think it's 2. I think it's just a single bond. Yeah, single bond, single covalent bond, which is really strong. That's the strongest of them all. Then ionic bonds are 2 charged ones that'll form. So acid and base. Acid and basic R groups, those amino acids, that group. Hydrogen bonds is polar. So any of the ones that have oxygen or nitrogen in them and have those partial charges, they'll make hydrogen bonds. Hydrophobic interactions are really, really weak. They happen from non-polar R groups. So where the R group has just mostly carbon and hydrogen, and they just hate water and they like to be with each other, kind of like fat molecules cluster together on the top of chicken soup. And then finally, another really weak one is van der Waals, and we don't really need to go over that because I don't think you need to know that. That's super weak, and that could happen between any of them as long as it's very close contact. That's when van der Waals forces happen. Quaternary structure then is a bunch of tertiaries like matched together. A lot of times it's four, and they'll link together. So we get like four tertiary structures making a larger globular molecule. Like, for example, hemoglobin. There's actually two alphas, two betas, or whatever. It doesn't have to be four. It could be more, it could be less, it could be two, whatever. But a lot of times, for some reason, it's four. And they link together with the same connections. They could have disulfide bridges, they could have ionic bonds, hydrogen bonds, maybe not so much hydrophobic and van der Waals because they're so weak, but the first three definitely would be coming into play for quaternary structure. So quaternary means fourth, tertiary means third, secondary obviously is two, and primary is one, first level. Okay, can you guys, I just want to take a look at your faces, well, whatever faces we have. Can you guys give me an indication of if you're following along with what I'm doing? Because I'm going a mile a minute here, and I'm not asking, like, I'm not giving you a chance for feedback. And I just want to make sure I'm not just talking to myself here. Like, are you able to follow at this pace? Okay, I
Yeah.
see thumbs up. If you can do a
For
thumbs up, that,
actually for mine, you keep on your screen share is like behind. I
oh,
don't know if
that,
that's for anybody else.
anybody else notice that? That could be a slow internet connection. Are you still outside on the Wi-Fi, or what's your deal? You're in?
No, I went back inside because my phone died.
Oh, okay. It might be a slow internet, but with that delay, are you still able to keep up, or is it
Yeah,
intolerable? It's
I
okay?
can just listen. Yeah.
Okay. All right. I'll also be posting this video. I'll post the transcript, or not but the recording on YouTube. I'll put the link up. All right. Okay, well, let's continue then. If you guys aren't dying right now and you're doing okay, my throat's still okay. Miraculously, you'd think I'd be dying right already, but still okay. All right, I'm gonna keep going. Okay, well, with proteins then comes the discussion of enzymes, and I'll just do a real quick and dirty, like just run through here of the terms, because literally we probably spend a week on enzymes. Like we did an enzyme lab, and like I could talk about hours and hours about enzymes, but I trust you can study this on your own. What you got to remember, oh, this is actually, the thing I did here is actually examples of proteins, enzymes, channels, carriers, receptors, defense, structural, energy storage, blah, 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 blah. This is the enzyme thing here. Sorry, I was in the wrong place. So for enzymes, what you need to keep in mind that there's an active site. All right, so if we have an enzyme, you know, whatever it looks like, there's some kind of active site. Obviously, the enzyme wouldn't really look like this. This is just a cartoon, you know, it doesn't look like a scary Pac-Man. But the active site is where the substrate binds. So the substrate is the molecule that gets acted on by the enzyme. It might get destroyed, or it could be like two substrate molecules get linked together into a larger molecule. Like some enzymes build, some enzymes destroy. Some enzymes just kind of convert things and change the shape of a molecule or whatever. But the substrate is what binds here then. So sometimes I draw that like a Dorito that fits in there. Okay, so that's the substrate. We could inhibit an enzyme. Like a competitive inhibitor might bind here at the active site. You know, if this thing binds here and it fits in here, now the actual substrate can't bind anymore. It doesn't fit because the competitive inhibitor has bound there. A non-competitive inhibitor might do something, you know, somewhere else. It binds over here and then changes the enzyme to such a degree where now that it bound over here, now the mouth is closed. It's not open anymore. It can't, because it changed the entire shape of this thing, and now the substrate can't bind. Why that changed shape all of a sudden, I have no idea. It's supposed to be a triangle, but okay. So non-competitive. So competitive inhibition can be overcome maybe by increasing substrate. If you increase the substrate concentration, the substrate, so the competitive inhibitor and the substrate will compete with each other. And the substrate, if you have lots of substrate, high substrate concentration, the substrate might sneak in there and might, you know, beat this guy out. So increasing substrate might help with competitive inhibition. But if I have non-competitive and I do this here and I shut this off, so this is closed permanently, I don't care how much substrate I put in, it's not going to work. So increasing the substrate will not work for non-competitive inhibition. That leads us then into cellular regulation of enzymes. So cells can actually control enzymes by turning them on and off. So inhibition is kind of talking about like when things go wrong and there's like a poison or something like that that messes with your enzymes and hurts you. This over here is talking about like how cells regulate their enzymes, and that could be, activating them or turning them off. So you could have allosteric activation where the enzyme is only active when this guy binds over here. So this is my activator, and that opens the mouth. Okay, my drawing is really bad on that. Sorry, kind of lost control of the tablet here. All right. So that it's just as bad. So that opens the mouth. Okay, so that's activation. Or it could be allosteric inhibition, where if this guy binds over here, it closes the mouth. It shuts it off. Okay, so then the cell can maybe turn the enzyme off when needed. Over here, it can turn the enzyme on when needed. So in this case, if the guy is not bound, like this is open over here, then the mouth is closed. Then the active site is not available. The activator binds allosterically, and then it opens it. All right. So that's that. Initial rate, that's like, on the graph, if you, okay, let me give ourselves a little bit more room here. If you're looking at enzyme activity graph over time, generally it looks like this. It kind of levels out. The initial rate is like the slope from here to here. So you do y2 minus y1 over x2 minus x1. That's slope. That's initial rate right here. When it's really steep, when it's straight, linear, and it's steep. And then when it levels off, that's obviously the enzyme is still working, but the substrate's all gone. The enzyme didn't like die or anything. The substrate just disappeared because it all got used up, like in the lab that we did. Oh, by the way, speaking of labs, I hope you guys noticed this, but I made a lab review document that goes through all the labs, even the ones we did not do. And I made videos for most of them, the ones that I consider the most important. I think I made 8 of them. So you need to watch those. They're like 10, 12 minutes each. So it's going to take you a while. It's like an hour and a half of videos. All right. But you still got a week. So it's not like the test is tomorrow. Initial rate, optimal conditions. So like, let's say you have like cold, and hot over here. Enzyme activity in the very cold would be very low. So this is activity. The reason it's low in cold is because there's low molecular, motion, and the substrate and the enzyme don't really interact. As it warms up, the activity goes up until it gets to an optimal point, and then it decreases again. And the reason in too hot is because the enzyme gets denatured, it gets destroyed by the heat. And the reason why that is because there's a lot of molecular motion, and it destroys the shape of the molecule. So we call this the optimal, let's say, temperature. All right, cool. All right, that's about it for proteins. Let's talk about any questions on proteins. Shout it out if you have them. Otherwise, I'm gonna keep going. Because we're 20 minutes in and I'm still on the first unit, so I gotta cruise now. Maybe I won't talk faster, I'll just say less. All right, carbohydrates, the next one. I'm certainly going to talk a lot less about them. They're made out of carbon, hydrogen, and oxygen. And by the way, proteins were made out of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. So that's kind of important to know the elements. Carbohydrates are just made out of carbon, hydrogen, and oxygen. And they have like a large amount of oxygen in them versus lipids, which have a very small amount of oxygen. So basically, the generic formula for a monosaccharide is C1H2O. So for every carbon, there's an oxygen, and for each one of these, there's 2 hydrogens. So it's like for every carbon, there's a water, and that's why they're called carbohydrates. For every carbon, there's 1 water, carbohydrate. Okay. So there's lots and lots of oxygen, which makes them kind of polar. They have a lot of polar regions to them, and makes them easy to dissolve in water. They love to dissolve in water. They love water altogether. There's monosaccharides, mono 1, that's just 1. Most common one we talk about most common ones are hexoses, like glucose, and you don't have to have the structure of glucose memorized, but it wouldn't hurt. Disaccharides are 2. So there's 2 of them, like sucrose and lactose and those guys that have 2 monosaccharides stuck together with a some kind of glycosidic linkage. Polysaccharides are many of them, lots of them stuck together in a chain. And examples are starch, cellulose, glycogen. Starch is basically like a polysaccharide that's a food storage thing from plants, and we use that for food. We eat that. Glycogen is like the same thing. It's kind of like starch, but it's in animals. So like our muscles and our liver stores sugar in the form of glycogen, long chains of alpha glucose stored in our liver and our muscles as a fast food supply, like a fast glucose supply. Cellulose is structural. It's made out of beta glucose. So these guys are alpha glucose. And cellulose is made in plants, not in animals. And it's structural. And that's what makes up wood and paper and cotton. And it's really, really strong. And the reason it's strong is because it forms these hydrogen bonds between the molecules that alternate. They go up, down, up, down, up, down. And then they form sheets and they roll up into fibers, which are really strong, which is kind of nice. So, and that's super useful to us because we make cotton clothing out of it and we build houses out of wood and stuff like that. Hexose just means it's 6 carbons, pentose 5 carbons, like ribose and deoxyribose in DNA. Glycosidic linkage is just the covalent bonds that connect them. It usually looks like C, O, and then C. Alpha and beta glucose. Difference is the orientation of the hydroxide on carbon 1. If it sticks, I got to think about this. If it's down, it's alpha. If it's up, it's beta. So that one little orientation, like if that goes down, it's alpha. Then we can eat it and digest it. If it goes up, it's beta. And then it flip-flops and makes cellulose instead of starch, which is crazy how that one tiny little thing makes such a big difference. Lipids are also only made out of carbon, hydrogen, and oxygen, but they have way, way, way less oxygen. They're mostly carbon and hydrogen, which makes them really nonpolar, super, super nonpolar, and really hydrophobic. And most lipids are triglycerides that are made out of glycerol and 3 fatty acids. They have that kind of structure like this. So here's your glycerol, carbon, carbon, carbon. Then there's an oxygen, and then there's a fatty acid. Oxygen, oxygen, and there's a fatty acid like this. So the 3 fatty acids stuck to glycerol like that. And most fats we can think of in oils like olive oil and butter and just about any the fat that's like makes us fat, you know, the reason I have a big belly or whatever, animal fat, any kind of fat like that is usually a triglyceride. Just about all of them are. I said already they're hydrophobic, they're nonpolar carbon hydrogen, da, da, da. Then another kind of fat is a phospholipid that makes up membranes, and they have that phosphate head, they have a glycerol, and then they have the 2 fatty acids. That makes up membranes, and a lot of times we just draw them like this. And we know that in cell membrane, they do the bilayer kind of spiel like this. Everybody knows this. This part is hydrophilic, this part is hydrophobic. The fatty acid tails hate water, they're in the middle, they're mostly carbon and hydrogen. And then these guys are phosphate that have negative charges on them that loves water. Cholesterol and steroids, that's a funny like 4-ring fat. It has that 4-ring kind of a structure. Okay, I can't draw it right now. Don't have enough space. Cholesterol obviously is in food. It's also important in cell membranes. It integrates itself in the membrane. You can go back to my PowerPoints and look at pictures of this. It's also the source of steroids. Steroids like testosterone and estrogen are made from cholesterol. So they're important for that. So cholesterol is actually kind of important for us. It's important in our membranes. It stabilizes the membrane, helps the membrane. Important to make these steroid hormones. The problem is if we eat too much, it can cause, you know, heart disease and problems with our arteries. It's unhealthy to have too much in our diet. All right, nucleic acids, you know, it's a DNA, RNA. They are carbon, hydrogen, oxygen, nitrogen, phosphorus. They're made out of nucleotides, so the monomer is a nucleotide, which makes polymers like DNA and RNA. Each nucleotide has a phosphate, a pentose, which is either ribose or deoxyribose, and a nitrogenous base, and those guys are adenine and thymine and guanine and cytosine. And we know that thanks to Chargaff, A binds to T and G binds to C. And we know that A and G are purines and T and C are pyrimidines, and these are small and these are big. These have 2 rings and these only have 1 ring. So, okay, I'm not going to draw it. It takes too long. Everybody clear on that? Good to go?
Yeah.
All right, let me see the thumbs up. Either that or do the button, you know, this one. Yay, I can do it too. All right, we're going to keep going. Yay. Cells. Oh, look, it's smaller. Yippee. Still a lot of stuff here, though. All right, you should probably know the organelles, like basic freshman or middle school, biology. The mitochondria is the powerhouse of the cell. You probably should know that, but you might want to know a little bit more about it, like how it does respiration and electron transport chain and ATP and all of that business. Nucleus clearly is important. It's surrounded by 2 membranes, so it's double membrane. It holds the DNA, it holds the nucleolus. The ribosomes are made there. It compartmentalizes these processes versus prokaryotes that don't have a nucleus. Our DNA is kept separate from the rest of the cytoplasm. Rough ER is associated right around the nucleus. It's responsible for making membranes for just about everything in the cell. Not everything, but just about everything. We call that the endomembrane system. It gives membranes to the nucleus. It gives membranes to the outer plasma membrane. It makes the smooth ER. It makes the Golgi. It makes lysosomes. So it is quite important. And it has those ribosomes stuck to it that make proteins that get exported. So proteins for export or proteins that stick in the membrane are made here. Smooth ER is like for detoxification and lipid synthesis. Golgi is packaging and processing proteins, modifying them. Lysosomes like holds digestive enzymes to break things down. The cytoskeleton is important. It's got 3 components: microtubules, intermediate filaments, microfilaments. I would, if you're like not going to study one, I would not study this one. I would know these and these. So microtubules are the biggest. They're hollow. They're real strong. They resist sort of bending. They're in flagella. They make up spindle fibers. They're important in transport inside of the cell, like vesicles get dragged along these tracks that are microtubules. So real big deal. They have that 9+2 configuration in flagella and cilia. And centrioles. Microfilaments are the smallest of the bunch. They're made out of actin. These are made out of tubulin, by the way. And they're important for muscle contraction. I don't know why I'm even writing this. I'm not going to be able to do it all. Muscle contraction, like cleavage during mitosis. Help me out. What else do they do? Muscle contraction, cleavage, and mitosis. Oh, cytoplasmic streaming. They carry things along inside of the cell. Bunch of important stuff like that. If I'm skipping stuff or you remember something else, shout it out. I'm just doing this off the top of my head here. Mitochondria have a pretty complex structure. We'll talk more about them when we talk about energetics. Chloroplasts are only in plant cells. They're responsible for photosynthesis. Peroxisomes, forget about. Cell membrane, phospholipids, lots of proteins in there. Sort of separates the inside and the outside, important for transport. The inside is hydrophobic. You should know about diffusion. You should know about water potential, and there's a lot of good water potentials practice problems on the math review. Actually, some pretty challenging ones, and there's a lot. So the only reason I did that is not because I think there's going to be tons of water potential on the test. I mean, there might be. I don't know. Probably not. But kids always get confused about water potential, so I gave you lots of practice. So anyway, if a cell's in pure water, it'll swell, and it'll like if it's an animal cell, it might explode. Cytolysis, we call that. If it's in salt water, it'll shrivel up because it loses water. Water diffuses out into the salt water because of the water potential issues, and we call that plasmolysis. And you should be able to explain this in terms of water potential. Like inside, the water potential is higher than on the outside, so therefore the water moves out. And the math review has lots of exercises on that. And by the way, I posted an answer key for all of it, where I wrote in the answers like this, it's on the Google Drive already, and I posted it on Classroom. You should know about mitosis. You can study mitosis on your own. I'm not going to go through all the stages of mitosis, but here's some extra like AP stuff that we didn't talk about freshman year. You should probably know what cohesins are. They hold the 2 sister chromatids together. You should probably know what sister chromatids are. You should know that they form during the S stage of the cell cycle. You should know about the cell cycle. Cyclin-dependent kinases, important to start mitosis. Without them, we can't get started. Cyclins activate the cyclin-dependent kinase. They form MPF, which is maturation promoting factor. You don't have to memorize that. Checkpoints prevent mitosis from happening. They stop the cell cycle in case something's wrong. Spindle fibers simply pull the sister chromatids apart. Cleavage is the splitting of the cell. Cell plate happens in plant cells where a new cell wall is laid down in the middle because it can't pinch. Mitosis is for growth. It's for repair. In some organisms, it's for reproduction, like in amoebas, they do mitosis to reproduce. In mitosis, if it goes haywire, if the cell cycle is messed up, you could get cancer because there's too much reproduction, too many cells form, you could get tumors. Good on mitosis? See one person nodding, good enough for me. Signal transduction was really complicated. There was a lot of stuff to talk about. I'll kind of shrink it down into a short summarized version here. 3 main things that can happen when a ligand binds a receptor. If you get a ligand binding a receptor, you could open an ion channel, you could activate a G protein, or you could activate an RTK. Oops, that's RTP, RTK. Those are the 3 possibilities. Ion channel, RTK, or G protein. Okay, so let's go with G proteins first. Let's say we get the ligand binding the G protein coupled receptor. That activates a G protein by binding GTP. The reason it's called G protein is because GTP binds to it. That could do one of 2 things. It could activate adenylyl cyclase, or it could activate phospholipase C. It depends on the cell, depends on what you need. Adenylyl cyclase splits ATP and produces CAMP, or cyclic AMP. Cyclic AMP is a secondary messenger, a second messenger, I should say, which then activates protein kinase A, which then kind of elicits some kind of a cellular response by phosphorylating other proteins, other kinases that then have some kind of a cascade probably that happens before the final response happens. Like, for example, glycogen getting broken down by glycogen phosphorylase, which gets activated by epinephrine, which follows this pathway right here. Another possibility is that phospholipase C gets activated, which splits phosphatidylinositol bisphosphate, my favorite molecule to say, PIP2. It splits it into IP3 and DAG, that's inositol trisphosphate. You do not need to memorize these names for the test. Just the abbreviations are good enough. Even that, they probably will tell you all this and then ask you something about it. If they ask a question about this stuff, you're not going to have this memorized, but understanding it ahead of time might be nice if they give you a problem with it. IP3 then opens up a calcium channel like on the ER maybe, or on some other organelle, which releases calcium into the cytoplasm, which activates protein kinase C. That activates some kind of a cascade to give you a cellular response, whatever that might be then. So adenylyl cyclase is the enzyme that makes CAMP. Phosphodiesterase is the opposite. It shuts down CAMP. It produces just AMP from cyclic AMP. So it's a way to turn off cyclic AMP so it doesn't just stay active forever. G proteins tend to have a single focused response. They're narrow. For example, glucagon and epinephrine use G proteins. Glucagon raises blood sugar. Epinephrine does a bunch of different things. It's neurotransmitter and gets you ready for like fight, flight, the autonomic nervous system. Another possibility is that RTKs get activated, and there's two separate monomers that join together to form a dimer. So we had this kind of a thing here. I don't know if you guys remember this. This is kind of how it looked. This was in the membrane here, and this stuck into the cell, and these are the kinases then that activate. These all got phosphorylated. I think these were tyrosine. These got phosphorylated and then did responses. So 2 ligands had to bind up here in the receptors. That's your RTK. To form this dimer, and the dimer then had all these spots down here that had kinase activity and made kind of a cellular response. That's an RTK, receptor tyrosine kinase. All right, this is a broad response. It's not as narrow, not as focused. You can activate multiple pathways. You could have like one thing happen, you could have another thing happen, all with one RTK getting activated. And examples would be like insulin or growth factors like epidermal growth factor, which turn on mitosis. Insulin does a bunch of different things, but it mainly lowers blood sugar by building up glycogen. All right, important concepts are amplification. So these complicated mechanisms that have all these steps, so many steps, very complex, you can amplify at each step. In other words, if you have like 10 of these, you could have 100 of these, you could have 1,000 of these, and it amplifies it and magnifies it. Kinases phosphorylate, phosphatases dephosphorylate. So kinases is an enzyme that sticks a phosphate on with ATP takes a phosphate off of ATP and sticks it onto the molecule. A phosphatase takes it back off and turns it off. In that chapter, we also talked about apoptosis. Oh, did I say it right? I swear I say it different every time. Apoptosis, apoptosis. Okay. Shout out with your mics turned on what this is. I need a break here.
When a
Cell death.
cell dies, it like.
Hey, you guys stayed
signals-
with me and you're still there and you didn't go to sleep. This is nice. Thank you. I would hate to have to listen to a guy rattling stuff off like this. So I'm sorry that you have to do this, but we got to do it. Also, I'd hate to be the guy that has to rattle it all off. Oh, wait, that's me. My life sucks. Okay, moving on. Any questions on signal transduction?
Nada.
I found that, I always thought that kids thought this was hard. Did you guys think that was a hard chapter?
Yeah.
Yeah,
It was all right.
Anyway, I know I did such an amazing job teaching it that it probably made it easy for you, but anyway, still might have been a little bit hard. Energetics, basically what I call energetics is respiration and photosynthesis. It's energy. It's how do we get energy? So in terms of respiration, it's how does a consumer, essentially an animal, take food like glucose and make ATP. Okay, so the respiration formula is glucose plus oxygen yields CO2 and H2O. What I didn't put in here is it also yields a whole crap ton of ATP. As a matter of fact, we could get possibly 38 ATP in aerobic respiration per glucose. Now that's hypothetical because some of the processes are inefficient. We actually get less than 38, but ideally we'd get 38. And there's a whole bunch of reasons why that is that we went into, which I may not go over right now. All right. I don't know if they're going to get into this. This is like chemistry mainly, not really biology, but a reduction reaction is when you gain electrons, and usually molecules get energized by getting reduced. So if a molecule gets electrons, it's energized, and so it gets reduced and it gets energized. So that's kind of weird. So NAD plus gets reduced to NADH. This is high energy. This is low energy. This is discharged. This is charged. It's kind of weird. Oxidation is losing electrons. So if we do that, we reduce the energy state of it. So NADH is oxidized to NAD plus. It's kind of weird. Anyway, so we start with glycolysis. So glycolysis just takes glucose and splits it into 2 pyruvates. And those pyruvates then feed into the Krebs cycle. Okay, and the Krebs cycle essentially gives us NADH. Now it also gives us FADH2 and a little bit of ATP. Not much ATP, but mainly NADH and FADH2. Those feed into the electron transport chain. And then the electron transport chain uses the NADH and the FADH2 to produce a lot of ATP. And that's where most of the ATP is made. So this is aerobic. That means oxygen is present, which means the electron transport chain needs oxygen coming in. It has to have oxygen. Without oxygen, this shuts down. Uh-oh, if this shuts down, first of all, we're going to shut this down. But second of all, this also shuts down. And the reason why is because in order to make the Krebs cycle work, you got to give it NAD plus back. And if you can't get NAD plus back and FAD back, this shuts down too. And if this shuts down, this builds up, and this shuts down too, which means you're getting, without oxygen, you're getting 0 ATP. But there's a trick that some organisms use, and that's anaerobic. With anaerobic respiration, this is already out, this is out. What happens is we got to get rid of these pyruvates, and we got to feed NAD plus back into glycolysis to keep that going. So what happens is the pyruvate is broken down, and it's either going to be made into ethanol or it's going to be made into lactic acid. This is getting really ugly over here. So what I think I'm going to do is give myself a little bit of space here. So basically what we have is this, is we get we're feeding back NAD plus over here to keep this going by breaking this down into either ethanol or lactic acid. And that allows us to give NAD plus back to glycolysis and to keep it going. It also gets rid of the pyruvate, so that doesn't build up. And that allows us to keep making ATP. So we get 2 ATP out of 1 glucose with anaerobic, which is very inefficient instead of 38, but at least the organism can survive, like yeast can do this in low oxygen conditions, et cetera, et cetera. We also do anaerobic by making lactic acid, but I don't want you to be confused. We don't do this when there's 0 oxygen like available to our body. We do this when we're doing aerobic respiration at 100% already. We're already getting those 38 ATP as fast as we can and as much as we can, but we're using all the oxygen we can take in to do this. So then we kick in a little bit of anaerobic in addition to this to give us a boost of a little extra ATP. And this is wasteful because it wastes glucose and it wastes pyruvate, et cetera, and it wastes NADH. And it builds up lactic acid, but it gives us a little energy boost so we can run a little bit faster or fight a little bit harder or whatever we have to do, lift a little bit more weight or whatever to get us past that emergency. And we can only do this for a few seconds, and then we have to get rid of the lactic acid. We have to take a break and we have to rest. And that's when you're sprinting really hard at the end of a race. All right. So do not memorize the Krebs cycle. Do not memorize the electron transport chain. Know the relationship of glycolysis, Krebs cycle, electron transport chain, and how glycolysis feeds the pyruvate into the Krebs cycle, how this produces NADH and FADH2 for the electron transport chain, how this gives back NAD+, that you need to know. This gives back NAD+. That's important. So anybody, when you study this, what I'm telling you is don't memorize the steps. Know the big picture, understand what's going on. And you probably should know that this happens in mitochondria. So you should know that, if you look at the structure of mitochondria, the Krebs cycle happens here in the matrix. The electron transport chain happens here on the inner membrane. There's hydrogen that gets pumped into the space, and the hydrogen diffuses back to the matrix, and it powers ATP synthase. This molecule that spins, and hydrogen goes through there, causes it to spin to make ATP. So that's kind of important. That I'd like you to know. And glycolysis happens in the cytoplasm. So glycolysis is out here, outside of the mitochondrion. So that means that pyruvate for the Krebs cycle has to get transported in here. That costs some, ATP also. That's why we don't get the full 38. There's other reasons too. The fact that NADH gives you 3 ATP and FADH2 only gives you 2, that has to do with where it feeds in. So like NADH feeds in here, FADH2 feeds in on the second step. So this pumps hydrogen more times than this does. So it gives you more hydrogen, more ATP. I would review energetics a little bit if I were you. Might be a good idea. Let's see. Aerobic, lactic acid, yada yada. Okay, photosynthesis. Same thing with photosynthesis. Do not memorize the detailed steps of photosynthesis. Instead, know that in the chloroplasts on the thylakoid membrane, that's where the light reactions happen. So if we have a chloroplast, here's the thylakoid, that's where the light reactions happen. So the light reactions are like another electron transport chain, but these guys are powered by light, not by NADH. Okay, and then the electrons move, la 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 la, and then they eventually end up forming NADPH. Okay, and in the meanwhile, hydrogen ions are getting pumped into the thylakoid. They're getting pumped inside, and those want to come back out. And when they come back out, they go through ATP synthase. That means in the light reactions, you're also making ATP. So you're making NADPH, which is P is phosphate. That's just like NADH, except a phosphate. And that's only in photosynthesis. So the P is like photosynthesis. And you're making these two ATP plus NADPH, which then feed into I'm going to do something. I'm clean this up a little bit. Sometimes in my haste, I actually end up. All right, so we got light reactions feeding the Calvin cycle, which sometimes we call the dark reactions. And what do they give? They give NADPH and they give ATP. Okay, the Calvin cycle then takes CO2 from the atmosphere with an enzyme called Rubisco, and it makes it into sugar. As a matter of fact, it makes glyceraldehyde 3-phosphate. That's a 3-carbon sugar, and that can get made into glucose then. Or into, beta glucose, cellulose, amino acids, lipids, anything you want to make really with this. So we call this carbon fixation over here. And the energy to do this, we get from NADPH and ATP from the light reactions. So sometimes people think that this happens during the day and then this happens at night. Nope, this also happens during the day because the only time the Calvin cycle can work is when the light reactions are working. So they both happen during the day. And at night, this shuts down. What am I forgetting? Oh, C4 and CAM. C4 and CAM plants you can review on your own, and you can watch videos on YouTube, or you can read about them in your notes. I don't think the College Board is going to ask you about those, but I think they're important to know about, like just for general biology knowledge. So maybe don't worry too much about them on the test. But they're plants that can handle low water supply and hot, dry climates. And they have tricks up their sleeve like spatial separation and temporal separation to get that carbon dioxide in, because Rubisco has that terrible flaw of binding oxygen and causes photorespiration, which is a bad thing, can kill the plant. So these guys have a way to solve photorespiration. All right, but we talked about those in class pretty thoroughly, so you can look at your notes. Hey, I got 10 minutes and I got 4 units done. Wow, okay. Let's take a look at where we're at. Oh, we got Lisa Buttergabe. Is that who that is?
Yep.
When did you join in?
At like 1:20 because I was busy doing something and I
Okay.
didn't realize.
I just have to decide if I'm going to count it as attendance. But if you came at 1:20, I'm going to count it because then you've been in here most of the time, right? I just have to do a quick screenshot because then I can keep track of attendance because the Zoom recording doesn't want to tell me names. How are you guys doing in terms of following along?
Good.
Doing okay?
Pretty good.
All right.
Yeah.
I might have to review the, what is it?
You can watch the video if you really enjoy people talking at 100 miles an hour. So anyway. Well, you know something? Let's go through genetics. And then molecular genetics, we just did that right before the shutdown. I mean, we just had our test on that Friday the 13th on molecular genetics. So you can sort of review that on your own. And that's what I did here. Because this was really late at night when I made this document and I was dying. And this is as far as I got. And I don't think I'm going to talk about it today either. But we can still later this week if you want to. I mean, we can do extra Zoom sessions too if you'd like to. All right, so genetics, let's see. There's so much to talk about there. You should probably know, well, you should know about meiosis before genetics, first of all. You should understand meiosis, how it reduces the chromosome number from, like in a human, it goes from 46 to 23. So it goes from diploid to haploid. And the way that works is homologous chromosomes find each other, line up, and then get split, and then get split again. So there's 2 divisions so that each cell only ends up with 1 copy of each chromosome, haploid. So that's sperm and eggs form. Males make 4 sperm, females make, well, that's in humans or animals, 1 large egg, 3 polar bodies. Other organisms don't necessarily do this, like plants or whatever, but the reason why is because the female has to supply most of the nutrients and stuff. So she makes 1 good, awesome egg and 3 junk polar bodies because the sperm doesn't want to bring anything. He just brings the chromosomes. So, all right. You should know Mendel's stuff. Like, especially like principle of segregation, that's pretty easy. That's the fact that they split from diploid to haploid. Like when you're making gametes, the gamete only has 1 letter in it. That means the 2 factors, the 2 genes or the 2 alleles get separated. Segregation just means separation. Independent assortment, kids get confused about that. That means that all the different chromosomes, if the genes are on separate chromosomes, they line up independently. They do not care about each other. And that means that if you have this parent, they're going to not just give you big A, big B, and little b, but they're also going to give you the combinations of the two, where, you know, the big letters and the little letters do not care for the A's and the B's. They do not care what the other one does, and the way they line up is random and it's independent. Okay, so you might want to draw that out for yourself and look back at your notes. Just make sure you understand independent assortment, because on the test they might say things like, these assort independently, without explaining what that means, and you're expected to know what that means. Now, if the two genes are linked, then you don't get all these combinations, because then there is no independent assortment. That's, if they're linked, all you get is these. You don't get these. That's because the two genes are on the same chromosome, and they do assort together because they're linked together. You should know what a test cross is. That's to determine if your dominant trait individual is big A, if it's heterozygous, or if it's homozygous dominant. And the way you do a test cross is if you cross this guy with homozygous recessive. You should know what genotypes and phenotypes are. Genotypes are the letters, the genes that are present. Phenotypes, how it looks. You should know how to do a Punnett square. As a matter of fact, you should know how to do every damn Punnett square in the history of the universe. You should know all about phenotype ratios, and I gave you a bunch of different phenotype ratios down here. You should know how to interpret those. Like, first of all, you should be able to generate the phenotype ratio from the Punnett square by counting the, you know, the numbers of individual, you know, offspring with different traits. But you should also be able to like interpret, like, let's say somebody gave you this phenotype ratio. You should be able to then figure out what the parents were, like what, you know, was their genotype? How did we get this? Obviously, this kind of a ratio is with two different traits. It's a dihybrid cross. You should know about probability. And by the way, let's look at some weird ones. So these are all like normal ones. These are all like the normal monohybrid, dihybrid. What about a weird one? Turn your mic on and tell me what this is. What the hell? 9 to 3 to 4. What is that crap? How do you get that?
Or a double heterozygous cross.
Yeah, but double het gives you 9 to 3 to 3 to 1. So this is really 9 to 3 to 3 to 1, because this 4 is really a 3 plus a 1. Why do you get 4 of the same? Like, why do you get 9 black mice? Why do you get 3 gray mice and then 4
Codominant?
white? Nope.
Epistasis.
Epistasis. It's epistasis. It's because the one gene affects if pigment gets put into the hair of the mouse. So if they have one dominant allele, if they're heterozygous or homozygous dominant for that color deposition gene, they're going to be either black or gray. That's the normal colors. If they're for that second gene, if they're little b, no matter if they're black or gray for the color, none of that black pigment or the gray pigment will get deposited, and they're going to be white. So you're going to have those guys. And on the Punnett square, that's going to look like this. So we're going to have these guys, 9 are going to be black. These 3 are going to be gray. And then this guy, and this guy, and this guy, and this guy are going to be white, because they're little b. So you got this extra, you know, spiel going on here. Anyway, so review epistasis. We practiced that a million times. I gave you that on the midterm. I gave you review problems for the midterm. Go over that again. Just make sure you're clear on that. This is another weird one. How would you, what? 12 to 12 to 1 to 1. What is going on there? And I'm going to slowly write the answer down, but try to shout it out before I write it down to impress me. 12 to 12 to 1 to 1. Someone, come on. I know people know this.
Isn't that like when the alleles cross?
Yeah, but the
I don't
two
remember what it's
genes must
called.
be,
Linked.
in order for them to cross, they must be linked on the same chromosome, right? So what you
Yeah.
have then is this. You got crossing over going on. Whee! And even though they are linked on the same chromosome, you know, like that, you get crossing over, and only a little bit of crossing over, only some crossover, and that's how you get these. So that means that the these gametes, only a few make these. Most of them are going to be this. Most of them are going to be this, which is going to give you the 12 and the 12. Only a few of them are going to be this, which is going to give you these guys. Okay, so then we can calculate map distance by looking at frequency of recombination. And by just saying, how many recombinant events did we have? 2 divided by the total events. Okay, so we'd probably get a number of offspring here. We'd probably get something like this, 7, 8, 121, 124. We'd add these two together and divide by the total times 100, and that gives you the map distance. And this you should know, and if you don't know, make sure you review that and go over that, because they've asked about this many times in the history of AP testing. They like asking about this. And man, if that shows up, you guys are going to get 5s because you guys know that so well. It's awesome. You should know how to do chi-square, although they probably won't make you do a chi-square calculation, but they might ask you to interpret one if they give you one. So make sure you know how to set up like the chi-square table and how to interpret it. And you should know about the null hypothesis. They love the null hypothesis. That's just simply saying the difference between this and, let's say I get this. Let's say I get 78 and 24. You say, aha, that's 3 to 1. But the 78 to 24 is not exactly 3 to 1. It's kind of different. So what I'm saying is that the difference between 78 and 24 and 3 to 1 are just simply due to chance. It's just a fluke. What I'm saying is this really is a 3 to 1, even though it's not exactly. That's what the chi-square is testing. You're affirming the null hypothesis. If the null hypothesis is true, if the differences are due to chance, that means it is indeed a 3 to 1. That happens if your chi-square value is smaller than the critical value. And the critical value you determine with degrees of freedom as number of phenotypes minus 1. You look up the critical value table at 0.05 and just go through a chi-square. I give you chi-square rundown in the math review too. Okay, so make sure you study that and learn how to do that. Odds are, once again, they're not going to make you do the calculation. Like you're not going to have to do the observed minus expected squared divided by expected, but they might ask you to interpret results, or maybe like a null hypothesis or something. Epistasis we already talked about. Some other weird stuff is polygenic traits. That's like if there's a bunch of genes for one trait, like skin color, for example. Multiple alleles is like there's more than 2 alleles. Sometimes there's 3 alleles, like blood type. We got A, we got B, we got little I. 3 alleles for one trait. So that's different than polygenic. Polygenic means a bunch of different genes. Here it's 1 gene, but 3 alleles. Environmental influences on phenotype, like the environment can influence, so like the pH of the soil could influence the color of a flower. Like I think it's a hydrangea is blue when the pH is basic, and it's pink when it's acidic. Sex linkage you should understand. Make sure that you incorporate X and Y chromosomes. Males are affected more because males they're hemizygous, they only have the one version. So you should know how to do Punnett squares with sex linkage, which are more tricky than without. And so practice that, especially if they give you a problem with dihybrid and they combine sex linkage, and if they give you something like this. If they combine like an autosomal gene and a sex-linked gene, autosomal is on a regular chromosome, sex-linked is on X or Y chromosome. Those Punnett squares get really tricky because you have to keep track of the trait. So, here you got your color or whatever it might be. Then you have to keep track of this thing, whatever that is, color blindness or whatever. And you have to keep track of gender. It's like having that extra, thing to keep track of with the gender. So sex linkage, yeah, make sure you study it. Make sure you go over it and study that stuff. Oh, linkage maps. Yeah, check out your linkage maps. Make sure you study that stuff. All right. This part it is 2:03, so this part I kind of don't want to talk about now. We can, if you want to, either we can do it today if you really want to, or we can do it later this week. I don't know exactly what I'm going to do in Zoom on Friday. Am I going to go over this exact same thing again or do something different? I'm thinking about posting this video. Then people who didn't come today can watch that. And then maybe on Friday we do something else, like go over the labs.